Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have started the hands on for the transcriptomic data analysis, and the goal is to identify differentially expressed genes among samples that come from different conditions. So, in this class we will be talking about sample specific bias correction. So, if you remember in the last hands on we looked at some of the preliminary analysis, we looked at the distance based correlation or heat map. Now, what we will do is we will actually generate a PCF plot. So, that is also part of the preliminary analysis, and then we will move into the sample specific bias correction. So, again here we are, right. So, we have started working with preliminary data analysis in the last class. We will complete that in this class, and then we will move on to the bias correction part. Once we have done that, we can go into the differential expression analysis, and then we will move on to visualization of the results, functional enrichment analysis over the subsequent classes. Okay, so let us move to R and let us complete the preliminary analysis part and see if PCA gives us the same data as the distance based clustering method, okay, and then we will do the bias correction. Okay, so let us move into the R terminal. Okay, here we are from the last class. This is where we ended, and we can now generate the PCA plot. Okay. So, one of the things I just wanted to mention is that we used Euclidean distance. So, first we did something called an R log transformation here, and then calculated Euclidean distance between the samples. Now, what R log transformation will do? It will reduce variability, right, that as we saw in our data for the genes that have low read counts, we reduce variability in the data. So, in that case we can use Euclidean distance. But if we want to do this analysis on the raw data counts, okay, we have to calculate something called the Poisson distance. Okay. Why? Because this raw data count there will be a lot more variability, there is no transformation done, right. So, we have to calculate something called Poisson distance, and we can calculate that using this library, okay, specific library built for this purpose, and we follow the same steps. Okay. So, we calculate this distance, okay. Instead of this function that we use this Poisson distance function. This comes from this library, and then we will generate the sample Poisson distance matrix, and we will use this p heat map as before to generate this distance based clustering or distance based heat map. Okay. So, I will go through this very quickly, okay, just to compare the results with our earlier results, right. So, and we will see we will get very similar results, okay, whether you do go through the R log transformation, okay, and through the Euclidean distance, or whether you go through the raw count data and use Poisson distance, we get the same results. Okay. So, I will simply copy this code because this part is same as before, right. So, instead of this RLD, right. So, we are just using this Poisson distance, although we are taking the names from the RLD dollar bin, but that is okay, right. We are just labeling the row names, right, with this bin and sample, but the data, the distance data comes from the Poisson distance. Okay. So, I will do, and then I will generate this plot. You can see there is a bit of difference. Okay. So, I will close this, and then I will generate it fresh, and you can see this data now. Okay. This is slightly different from before, okay, and again we have this group names and the sample names. Okay. So, you can see this G1 S5, G1 S9, these are the sample names, and again you notice the same thing, right. These G1 samples are very close to each other, right. So, darker color means smaller distances, and white color means larger distances, and here the distance is in terms of counts, right. Okay. And as you can see, the rest of the groups they cluster together very nicely, like G2, G3, G4, and as before there is an outlier G4 S60 in this sample looks like an outlier. It does not cluster with this group here or with the G1 group here. Okay. So, this gives the same results. So, what we can do now, we can go into the PCA, right. So, we can generate this PCA plot. Okay. So, to do that we again have this RLD, we can actually go with this RLD data, right, and this R log transformation that we did, and we can use this function called plot PCA. Okay. So, this will generate this PCA plot okay, from the data, and it will give these PCA percentages as well. Okay. So, this is a very easy way to do this, and it will tell, it will also label based on this bean and the sample. Okay. So, let us try this command first, okay, based on this R-log transform data. Okay. And to see this, like we see this PC1 here, and PC1 explains about 63% variance, and then you have PC2 on the y axis, it explains about 14% variance, and you can see these G1 groups are around here, right. So, the samples from G1 group, they are around here, they are clustered together in this PC plot, and the other groups are clustered around here. As before, you notice probably G4S16 is somewhere here, right. So, from all this analysis, it is clear that we should perhaps discard S16 from our analysis, because it looks like an outlier. Okay. So, this is something we can do, right, and we can come to this conclusion based on these two different preliminary analysis. We can also do this slightly differently. Okay. We can generate this PCA data, okay, and we can calculate this percent variance, and we can use this ggplot library to actually also plot the PCA data. Okay. So, we generate this PCA data from this plot PCA function, right, again using the R-log transform data, right, and this will store this data inside this PCA data variable. So, we can run this first command here. Okay. This will do the PCA and store the results in this PCA data variable. Okay. So, this is done now, right, we have stored this, and it will also calculate this PCA data percentage variation, right. So, percentage variation that is explained by this PCA data. Okay. So, in our PCA data, when you do PCA analysis, you might have 10 PCs, 100 PCs, right, and for each of them you will have this percent variation, percentage variance calculated or explained by that PC. Okay. So, we can simply store this, right, we are just simply converting this as a percentage from this PCA data analysis. So, if you look at this PCA data component, right. So, again you will have these different components PC1, PC2, group, bean, etc., right, and from this PCA data we are taking this attribute percent variance. So, this is the percentage of variance that is explained by each PC, and we are storing inside this variable. Okay. So, now we can load this, use this PCA data and percent variance to actually plot this. Okay. And you will see we will get a very nice plot, because we want these groups to be colored similarly, right. So, you see this here in this plot, it is okay, we can say okay, these are the different these are the group 1 samples on this side, but maybe if we can color them with the same color, it would be actually better, right. So, let us try that with this ggplot2, right, and the commands for doing that is this ggplot. We take the PCA data that we generated, okay. The x axis and y axis will be PC1 and PC2, these are the first two principal components that explain the largest amount of variation in the data. Color equals to bean, right. So, color the points according to the bean, right. So, part of which group they are, and shape of the points should also be according to the bean. Okay. Then you have this plus g of points. So, point size of these data points, then the x lab, the level of x axis, right, and it should say, it should read PC1 percent var 1, right, percent var 1, this comes from this variable percent var. Okay. So, this is the percent var 1 means this is the variance explained by the first PC. Then similarly have percent var 2, this is the percentage of variance explained by the PC2. Okay. So, these are simply referring to the x level and y level, right, the level of the axis, and then you have the coordinate fixed. Okay. So, this is if you are not familiar with ggplot, do not worry, you can go and look up a bit this ggplot2 manual, and you will see this is how we can specify all these axis, the point size, etc. It's a bit different from what we have used before, right. So, for plot function, we can inside this plot we mention, okay, where x limit equals to c something, right, y limit equals to something, the x level, y level, everything we mention inside this plot function, right, in our, but ggplot is different, right. So, we have this plus and then whatever you want to add we just add this with this plus sign. You can see this for all of them, right. So, if you have this geometry point, you have this x label, y label, and so on, and you can of course add a lot more to this, and you learn that, right, by exploring these functions. Okay, so we now run this command, so and see if we can get a nice PCA plot. What is happening? Okay, no, cancel. Okay, so what I'll do is then I think the copying didn't work, so I'll just copy the code again, right, and I'll run this code here, and you can see now this is actually better, right, because each group is colored with one single color, and then also the shapes are different, right, and again this one sample is an outlier here, which we may have to discard before we actually go into the differential expression analysis. Okay, so I think this preliminary analysis actually gives us a very nice idea about which samples to take for analysis and whether there is an outlier, and it also now tells us, okay, G1 is the interesting group, okay, because it stands out from the rest of the samples. Okay, so if we are going for differential expression analysis, we should be considering G1 versus rest of them, or maybe G1 versus G2, or and so on. So, we have now finished this preliminary data analysis, and we can now move on to the bias correction part. Okay, so we have discussed about different types of biases that we can see in RNA-seq data, and we can try to at least identify or minimize some of these biases. Okay, so that's what we are going to do now in the next step. Okay, and for this bias correction part, we'll use this package called EDSSEC. Okay, so again, I'll show you how this package looks like. This is a part of bioconductor. So, we can simply search here. Okay, so you can see this first result, bioconductor EDSSEC. Okay, we have talked about this very briefly in the theory class, right. So, here again it can do this adjust for GC content, gene level effects on read counts. It can do different types of methods. So, it has like at least 3 methods that it can do, right. It can do a lowest regression or global scaling or full quantile normalization, and it can also do between lane normalization, or means between sample normalization. Okay. So, we'll just take the within lane normalization, because we want to correct for the biases, right. These are sample specific biases which we can correct through the within lane normalization. Okay. So, this is what we are going to do. We will try with the GC content normalization, and we will first have to install this package. Let us see if it is there or not, right, and then we can if it is not there, we can then try to install it, and then we will run the codes to correct the data. Okay, so let us remove all the figures etc. that we have generated, and just also clear the window, right, and then we can start afresh. Okay, so library EDSSEC here. Okay, so it's installed. Otherwise, you can simply install with this command line using the BioC manager, and then we can now use this EDSSEC to do the bias correction. Okay, so we'll try this for the GC content. Okay, and we can again look at the manual and all the functions. Okay, that's are there. So one of the things we need to do is this within lane normalization, right? This is the function that we will use for our bias correction part, and you can see again we need to create a data format, specific data format called expression set. Okay, so I mentioned, right, for many packages you will see they require a specific data format to be created from the count data or matrix data that we are working with. Okay, so we'll do that first, and then we'll run this within lane normalization, and then we'll export the data. Okay, in a text file or a tab delimited file, as we have been doing, right. We have seen this count data in a tab delimited file in a text file. We will do that after we do the bias correction, and we will work with that for differential expression analysis. Okay, so let us try this part. Okay, and there are a lot of other functions as well, and you can see some of these functions are also overlapping with other functions. For example, you have the plot PCA, you have plot RLE. So we'll talk about plot RLE maybe later, we have used plot RLE, like I have shown you some RLE plots before, but we can also use this later on when you do the differential expression analysis to look at the normalized data, etc. Okay, so again we can go to this within length normalization method, or maybe seek expressions set part, right. This is a class of data right, that we need to create from our count data. Okay, and again this is mentioned how you can create this, right, what kind of attributes or data methods that you need to include in there, right. So, and there are some examples how you can create this, right. So, the function is new sec expression set, right. So, we have to give the counts, then we have to give the pheno-data variable, and then we have to generate this and assign this into this data variable. Okay, so, we will do that, and then once we do that, then we need this within length normalization methods function. Okay, this will do this normalization function, and you can see these different methods that can be used for doing this within length normalization or bias correction, which will give you that, right. And then x is the data that we want, right, and y is the variable for which we want to do the bias correction, right. So, we will take the example of GC content here, and we will do the bias correction for GC content in our data. Okay, so, let us move on to our data, okay, and then I have again prepared a set of codes that we can use. Okay, otherwise it will not be possible to complete this within this class. If you want to search the codes and then search the commands and set up everything, that might take quite a bit of time. Okay, so here it is, right. So, we are loading this data, right. This is the raw count data that it will take, then it will take the library, right. So, we will load the library EDSEC, then we will load these two data sets that are part of this EDSEC library. Okay, so, it is GC and is length. Okay, so, one thing I should mention now is that the data set that we are working with is an East data, okay, Saccharomyces cerevisiae data. So, we can utilize this East GC content East length data. So, this East GC East length is very simple, right. Is what is looking at is the GC content of the genes as well as the length of the genes, right. So, we need this GC content information if we want to normalize based on the GC content. We can also normalize with the length data, but for this purpose, for this hands on, we will just focus on the GC content based bias correction. Okay. So, let us run this. Okay. So, the first thing is we load the data afresh, right, in this data variable. Maybe we have loaded already, but I just want to run this again, make sure we have everything in order. Okay. So, this data is here. The next part is to load this library and this data, two data sets. Okay. So, first we load the library DSEC, then we get this two data that we will also load. Okay, data is GC and is length. Okay, is length is probably not required for this hands on, but anyway we can load library EDSEC. Okay, and then these are the data that are present in the EDSEC library. Okay, so we can simply load the EDSEC data, and that we will use for our analysis. Okay, so the next part is actually creating all the data format, the right data format. Okay, as I mentioned, we want to create this data format, which is the sec expression set before we can run this within length normalization. Okay, so you can probably see these two parts, right. The first part is looking at the intersection between the row names of the data, right. So, what are the row names of this data that we have loaded? These are the gene names, okay, and the names of the East GC, right. So, this East GC data also has names, and these names are gene names, okay, and we are take the only the intersection, right. So, wherever we see the common genes across these two data sets. Okay. So, this is the command sub intersect row names data names is GC. Okay. So, here we are, right, and we can find out, right. So, these are the gene names that are common. So, this is a large majority, as you can see, out of 6,800, about 6,700 they are present in both these data sets. Okay. So, that is good, right. So, we have found most of the genes in these two data sets. Now, what we are doing now is we are creating this matrix for taking only these genes. Okay. So, this sub is the list of genes, right, and we want only these genes and their count data for the bias correction. Okay. So, this is what we are doing here. We are taking this data, part of the data, right, a subset of the data, and then we are converting this in matrix and storing it in this math variable. Okay. So, math will have this data now. Okay. As you can see, again we have this sample, we have the genes. The only difference is now we are working with only a subset, right, which are common across these two data sets, right. So, because we cannot do GC content correction if we do not have the GC content data for any gene, right. So, that is why we need to work with only the intersection between these two data sets. Okay. So, now once this is done, right, this is we are ready to create this new sec expression set. Okay. Again, it might seem a bit complicated, but we are following the manual. Okay. Exactly what is given in the manual, we are following that. Okay. So, what we will do is we will create this new data variable, right, and we will assign this new sec expression set, right, that we create into this variable. Okay. So, the first part here is the math, right. So, this is the matrix that we have just created, this is only the count data. Then you can also define this normalized counts variable. Okay. So, this normalized counts variable, we are just data is in matrix, right, but there is no data now, it will be created. The number of rows will be number of rows same as the matrix. Okay. So, matrix has let us say 6700 rows. This normalized data matrix, normalized count matrix should also have the same number of rows, right, the same number of genes. Then you have the n column. So, number of columns should be same as the matrix file, because we have 16 samples here, also we should get 16 samples, and the dimension names will be same as the matrix. Okay. So, everything should be same as the matrix, only the data will be different, that will be generated after the bias correction. Then you have the pheno data, which is given as the annotated data frame, right, which actually gives us the conditions or the factors of this experiment. So, as you can probably see that the first 4 samples belong to group 1, the next 4 samples belong to group 2, the next 4 belong to group 3, and the final 4 belong to group 4. So, this is what we are mentioning when we are saying this factor C, etc., etc. Okay. Then you have the row names, right. So, this is the row names given for this data frame. This will be data file, right, that this data variable that you have loaded, the column names of that data, right. So, these are the sample names, which will be the row names in case of phenotypes, right. So, pheno data, and then you have the feature data, which actually belong to this each GC sub. Okay. So, this is based on the GC content, right. So, we are looking at GC content here. So, this belongs to the each GC sub. Again, it might seem complicated, but again we are following the functions that are given in the manual, and I will show you in a moment. Okay. So, then we can run this in the, it is a big command, right. You can see this in it goes to multiple lines, but in R it is okay, right. It simply see this plus, right, and then we can run the full command. Okay. It has generated this new data now, we can probably type, and it will show you, right, this is the sec expression set class of data. Okay. This is what we wanted, right. This is what we want to create before we can run this within length normalization method. And again, you have some statistics about the data, what are these assay data, how many samples, etc., sample names, etc., all these things, the feature names, these are all given here. Okay. So, what I wanted to show you is that we are running this according to the manual, right. So, where we create this new sec data method, right, and you can probably see, so we have this normalization. Okay. So, I will show you here perhaps in one of the examples you can see this clearly, right. This data is created like this, right. You can see this, right. So, we have just replaced this with our experimental design and our data. Okay. And then we are following the GC content, right. So, we want to correct for GC content bias. So, that is what we are going to do after we have created this data set. Okay. So, the next step is to actually run this within length normalization, whether and then use a specific type of method. We can use lowest method, we can use full quantile normalization, right. And then, so there are 3 methods that are mentioned. So, we can use any 3 of them. Again, these options are given on top, and you can find them, and then we will create the normalized data that will be created. Okay. And we can then export that normalized data into a text file. Okay. So, let us do that now. Okay. So, we have created this new sec data, right. So, this is stored in this variable new data, okay, and we now want to do this within lane normalization. Okay. So, that is the goal, that is the next step, which is this normalization part. Okay. All right. So, let us run this. I have copied this. So, we can run this command and we can probably see one thing, right. So, we are this is the data, right, where we are running this normalization within lane normalization. Based on GC content, we are running full quantile normalization, round the values to true because we want these normalized counts to be integers. We will load them in dsec2 which expects raw counts, right. So, that is why we want to say round equals to true and offset is false. So, there are two options in this package, that you can keep the counts as the same as the raw count, and then you can set offset values which will give you the normalized values. But in our case, we just want to create these normalized counts which we will use for the differential expression analysis. Okay. So, we have run this now and we can say norm, right, and the data is here. Okay. So, how do we actually look at the bias? Okay. So, whether there is any bias or not. Okay. So, you can see this by using this command bias plot. Again, this is part of this package. There are other plotting functions also, and you can use this, and this will give you the bias plot. So, this is the GC content and the gene counts, right, based on this bias plot. Okay. So, of course, gene counts cannot be negative. So, there is some sort of scaling here. Okay. So, this is how we can check this. Now, what we will do is we want to export this data, right. So, to export this data, I will use this command norm counts norm. So, it will get the normalized count data from this normalized data set, right. So, this norm data, this is the normalized data, and from norm counts, if you use norm counts on norm, we will get the normalized count. Okay. And we want to store this as data frame inside this variable norm2. Okay. And in the next step, we will write this norm2 variable using this command write dot table, right. This is a very similar command as read dot table. So, read dot table will read the data from a text file, whereas write dot table will write this variable data, this matrix data in a file, and you can say you can mention this file name, right, and then you can tell how it should write this. Okay. So, this sep equals to /t means you should separate, it should separate the data, every column using tabs. Okay. So, we want this tab separated file. Quote means f. So, if you do not use this command, you will see this, all these values will be within double quotes. So, we do not want that double quotes, right. So, we will just write, so quote equals to false, right. So, f just stands for false. Instead of writing the full thing, we can simply say f, and it means we do not want any quote when we are writing this data. Okay. So, we will run this, right, we will get this data normalized count data, okay, and we can check little bit normalized. Okay. Maybe we can try head norm2, right, and you will see this is different from what we have seen earlier. So, from the raw count data, this is actually different. You can compare, okay, and it should be different because this is corrected for the GC content bias. Once we have generated this data, we can simply write this table, okay, and we can use this command write dot table, and this bias corrected file will now be generated. Okay. And we can verify this through the command line. We will have a look at that in a moment. Okay. Before I look at that file, I just wanted to say, like, we can also look at this within lane normalization, whether this normalization has happened, what kind of normalization has happened, what kind of adjustment has happened using the RLE plot. Okay. So, we have used this command before, and here also within the package EDSEC we have this plot RLE command, and we can look at the data before normalization, which is the new data, right, and also look at the normalized data, which is the norm variable, right. So, we can use this plot RLE new data and plot norm, and we can see very quickly, right, how this normalization changes the data. Okay. So, this is what we are going to do, and I will just move this little bit so that you can see the data. Okay. So, these are the S1 to S16 samples, and you can see this is before normalization, right. And after normalization, you can see there is some difference, right. Again, depending on the type of normalization, this will change. Okay, you can see the distributions have changed a little bit. Okay, so this is what bias correction will do. Then we have corrected only for GC bias, and of course we can take other examples. You can do this for other methods. One final thing we want to see is that we have created the bias corrected file. Okay, so this code is complete now. We have run this code. We can come out and we just want to see that we have created this bias corrected file. Okay, so here is the bias corrected file, right, and we can open this with VI and just to see again that we have created this file and saved it in the right format. Okay, because we will load this data next when we do the differential gene expression analysis. Okay, so to summarize, we have completed the preliminary analysis and we have seen, right, whether the samples are correlated or whether there is an outlier in the sample based on two types of analysis. One is distance based clustering, the other is the principal component analysis, and both gave us very similar results, right. So we get some very useful insights into the data before we actually jump into the differential expression analysis part. And then in the second part, we have looked into the bias correction. There are different types of biases that are present in the data. In this hands-on, we simply looked at the GC content bias and we have corrected for that bias, and we have generated this bias corrected data that we can use now for the differential gene expression analysis. Thank you.